you announced the name and other thing?
Yeah, I have announced.
Okay, fine. Okay, so today the session is regarding the plant tissue culture of orchid plants. Orchids are basically very useful and commercial commodity in terms of the floriculture, and there are millions of plants are being exported to the developed and other parts of the country of the world. And second thing, it is used for the as ornamental in the floriculture business. And second, this orchid, it is a medicinal value also, and it is being used in medicine for the alternative system of medicine. So for these two reasons, it is has a very huge commercial importance. And but thing is that it is like high altitude plant, and it is growing at a low temperature. So if we want to produce this plant in the plains where temperature is high, so we have to maintain this in a tissue culture lab where the artificial climate is being created. So for this, basically, plants are grown in a tissue culture lab, and for this tissue culture lab is not like a small one. It is a commercial tissue culture lab. So where,
Dr. Ajay,
yeah,
you're not started the video, or after explaining a bit, you will start the video?
yeah. After that, I will start video.
Okay. Please continue.
Yeah. So basically, for this, because this is a backdrop I want to give. So once like the tissue culture lab is being made, and that is a commercial tissue culture lab, where you produce the millions of plants to meet the market demand. And the market is like a global market, as I told. So the basic requirements are the same in any tissue culture lab, but it's a plan, organization, and the productivity protocols are entirely different, because we have to produce millions of plants. Accordingly, the protocol changes. And in this case, the lab is being planned that way. So there are the three major steps for a tissue culture. One is a mother plant, which is called the X plant. So we obtain it from the area where they are being grown in a natural habitat. Number one. Then they are brought to the lab where they are being prepared for the tissue culture process. And in that process, there is called the process called surface sterilization. We have to remove all the contaminants from the surface present on the X plant. Then we prepare the media, which that is called, basically it is a food for the plant which is grown in the lab, and that food is called media. So media is very specific for this orchids. So once we prepare the media, that media is basically provides the nutrients for the proper growth of the plants. And once the media is prepared, then it is, because this media is in the environment, so it has the contamination in the water and in the containers. So we have to remove the contamination by process of autoclaving. And once it is autoclaved, so autoclaving is done at a definite temperature, that is called 121 degree temperature, 15 pound pressure, and for a fixed time, that is a 30 minutes. So once this is done, so the media is being sterilized. Now other side, as I explained, that the surface sterilization is also done on the X plant. Then we take it to the aseptic platform. That is basically laminar airflow, which sends the sterile air, and we work on that area where the process is being carried out. We have to, like, first we have removed the contamination from the X plant. Second, we have removed the contamination from the media. Third, we are removing, or we are working in an aseptic platform where there is no chances of contamination. So in that area, aseptic area, the X plant is being transferred into the culture media. This we call it as an inoculation. So once inoculation is done, then that media is kept in a climate controlled atmosphere. So there are three things. One is the temperature. So for orchids, temperature is 20 degrees Celsius plus minus 2 degree. Lights, lights are being provided artificially by the fluorescent lamps, and it has a particular intensity, luminosity, and then humidity. So humidity is already maintained inside the flask, and initially the humidity is around 95% RH. That is called relative humidity. So once the plant has gone, explant has gone into that flask, and we transfer those flasks, containers, all those containers in the growth room. So where these conditions are met, the light, temperature, and humidity. So there they are being kept for happens for the sake of speed. Some people get, when business people, they establish the labs, so they want very fast productivity. So but it is worthless, because plant will grow at their own speed, and initially it takes around 3 months to establish the culture. So once the culture, that means the plants, they can keep multiplying inside the flask, that we call it culture. And then once we have sufficient number of cultures, so then we do the next process, that is called subculturing. Suppose we inoculated 1,000 plants, and in that media, these plants will multiply around 20 times. So 1,000 plants will become 20,000 plants within 3 months. Now those 20,000 plants need to be subcultured. Again, we have to separate them and put into the another freshly made media, which already we have made. And once it is done, subculturing, then the cycle goes on. So in this first step, subculturing, the plants, the aerial part is growing. That aerial part is called the shoot. So they are growing and they are multiplying. Now, because only shoot cannot grow or be transferred to the land, so we have to initiate the rooting in those plants, and that is called root induction. Then we prepare another kind of media where these shoots are being transferred for the rooting, and then roots are being roots are being developed. They again take around 2 months, around 8 weeks. And once the roots are developed, so the plant is now ready. Now plant is ready inside the flask, but they are still not outside in the environment. So to transfer those plants in the environment, we take out the plants from the flask and transfer into the tray. Those are the common tray being used in the nurseries. So that tray is basically containing the media, hydroponic media, where the nutrients are required, are already present, and those nutrients, slight nutrients are there. And once they are like agar, because there is a media present in the roots in the plant, so those are being washed, and then after washing, they are transferred into the trays where the cocoa peat and the hydroponic media is there. Now there we this tray we acclimatize for outside temperature within the lab itself, and once they are acclimatized, we transfer to the greenhouse. So in the greenhouse, the further humidity is being reduced, and then light intensity increases, and plants become hardened. This process is known as the hardening. So once these plants are hardened, then from there, from the trays, they are being transferred to the pots. So those pots are normally like 4 inches pot or 5 inches pot. There they are transferred, and they are ready for the transplantation in the field. So on this 4-inch pot, the plants are being sold for the commercial production of the orchid plants. So this is like the process up to the hardening. So once the plants are hardened, then they are ready for the outside growth. So this is the basic procedure that I explained, and now I will show you the proper process, the in the lab real time, how this is being done, how it is being how it is being done. So just give me 2 minutes. I will just open the lab, and then I will just transfer the take you to the this the real show how it is being done.
Okay.
Just so I'll just log in again. So just give me 2 minutes.
Yeah. Yeah. Okay.
Hello.
Hello.
Hello. Yeah.
Yeah.
Am I audible?
Yeah, you're audible. I can see you. I mean, I can listen to
Okay.
you.
Okay. So, so now, like, I'll be showing the lab how it works. So the people they can have a real how it is happening in the real time. So can you see? Hello. Video is visible.
Yeah, it is visible.
Okay. So this is our uh, this R&D lab where we do the this entire floor is for the tissue culture. So once we enter, so there is a this air curtain. So there is an air curtain that basically stops the excess air and dust coming inside. And then this is a airlock room. So where the sleepers are removed, juices are removed, sleeper inside board, and then we enter into the media room. So this is a media room. So this is the media room. So we have different kinds of nutrients, the chemicals which are being used to prepare the media, that is nutrition for the plants. And then using a certain balance and the pH meter. So the media conditions are set. And once it is being done, so for this, the refrigerator is there to store the media and the chemicals which are like heat sensitive. So we have to store the media there, and we use the RO water, that's a highly pure water. And for media melting, we use the microwave. So this is a media preparation room. So flasks are being made. We make the small phyta jar, the tissue culture jars, and then they are the media is sent for the autoclaving, that is in another lab. So once that media is ready, I'll show you how this media. So now this is a inoculation room. So we are entering into the inoculation room. So here you have the different aseptic chambers. So these are the aseptic chambers. So this is the our inoculation room. So what we do, so you can see, so these are the media basically made. So this way the media is now first it was made in the media room, and this is a sterilized media. Now you can see there is no any, it is made on I think 1 week back. So still it's all clear, so no contamination. So this we have to ensure. So once media is ready and stored for 4 to 5 days to check whether there is a contamination, and once it is done, so then this real process is being done here. So this is a laminar flow, and it is 0.02 micron filter. It is sending the sterile air, and so these are the sterilized media, and they are being, so this aseptic thing, this work is very critical in fact. So once the media is ready and the cultures are ready, so we have to work under the aseptic conditions. So there is a glass bead sterilizer where we sterilize immediately, instantly the forceps and the these scalpels are being sterilized, and then this process is being done. So same process. So this process is going on. So this is the aseptic technique. So once this is done, so then the plants which have been transferred, all the culture. So now you can see these are the, we have to keep them for some time here, and here you see, earlier they are kept like this, very thin population, and then once they grow, you can see the dense growth. So this is like one plant is multiplying 20 times. So if you are like having 1,000 plants, so they become 20,000 plants within 3 months span of time, around 12 weeks time. So after this process, then they are being transferred to the growth room. So this is a growth room, and here the cultures are kept. Here the multiplication goes on, and depending on the time period, so these plants are being grown. And this is like, here we do the shoot culture, and then we, as I explained, then we have to transfer to another media where the rooting is being done. So then plants shoot and root are ready, and after that they are being transferred to the tray. So I'll show you how the, you can see, if you can see here, you see, so roots are visible, so roots are being developed, and once the roots are ready, so then they are transferred to the for the tray. So like this, so there are different conditions they are being kept. So this we have to monitor accordingly. And here see the lights, different cells have different intensity of light depending on their requirement. And there are the air conditions, so alternate air condition day and night, because we have to maintain the temperature. And the humidity is maintained within this site. So light is there, temperature is there, and the humidity is there. So this way, so once they are multiplied in large number, so then same process there we have to take like this, and then we do the subculturing. So again, so this cycle goes on, culturing, subculturing, culturing, subculturing, and then finally, once the roots are developed, so those roots, with roots, the plants are transferred to the tray, and after that they are sent to the greenhouse for hardening. And once plants are hardened, so they are ready for the sale. So this is the some brief process and the aspect of this for the sugar cane now. So we'll be getting, this is a one specific variety of sugar cane. So we have to produce 20 million plants for our buyer. So this variety will be produced in large quantity and then will be transferred. So similarly, the different like another plant is a ornamental, this aglonema is also under production process. So depending on the market need, we are doing this tissue culture. So orchid we have huge order, so that is why this we have started this one. So I think with this I should finish, and if anyone has any questions, so I'll be happy to answer those questions. Yes, Rajni.
Thank you, Dr. Ajay, for the presentation. I'll now take up some questions. So the first question is orchid cultivation popular in India? Where are orchids mainly grown?
Where are?
Is orchid cultivation popular in India? Where are orchids mainly grown?
Okay. So as I told, orchids are, they are the cold climatic plant. So they grow in northeast of India and in Himachal Pradesh, Jammu and Kashmir. So these are the area where the orchids are naturally grown, and there are certain companies they are producing, especially in Pune and Bangalore, they are producing and they are exporting also. And we are also on the same line. So we'll like we have a lot of culture now. So once the process is over, so we will be sending to those buyers. So it is done. It can be done. It's not a big deal, because technology is the same. Only we have to change the species.
Okay. And next question is, what are the new advances in plant tissue culture of orchids in recent years compared to 10 to 15 years back?
Yeah, this is like basically it is a research based question. Someone must be working on the research of the orchids. So what happens basically, we as I told you, it is being used for the two purpose. One is for the floriculture industry, where the orchids are being sold, and for this market we have to keep on bringing the new varieties, new changes, new phenotypic changes in the plant, so that the something new comes to the market. So that is the innovation, and that is through the molecular breeding. It is done through the molecular breeding, and then soma clonal variations are there. So we select those new type of the characteristics in the plants, which will be new for the market. So and then for the secondary metabolites, the orchids are being used as they are used in the medicinal, they have the medicinal importance. So those secondary metabolites are being extracted by the either by the fermentation technology or by the suspended culture method. The different kinds of processes are there. So we can produce the secondary metabolites, which are ultimately purified and then used for the as a medicine and for other substances. Yes, please.
Okay. Next question is, please tell us about your interest. What made you to take an interest in agriculture research?
Yeah, this is a very important question, in fact. So, the agriculture in India from centuries is being like traditional agriculture, and the farmers what they are getting is not the relevant to their labor, and the cost of production is increasing. So what the agriculture basically, it comes under agriculture biotechnology, where we can produce the crop, high value crops, and those high value crops will give the farmers a better return. Like you see in market, like you might have seen the see the one best example is banana. Banana is now almost 90% banana is through the tissue culture method. Then pomegranate. You see the pomegranate in the market, same size, same color, same flavor. So that is by the tissue culture technology. Papaya, same shape. You might have seen same shape of papaya. Millions of papaya they are coming from truckloads of papaya. So that is when we maintain a specific characteristics of the plant, and in a huge quantity with the less number of diseases and better productivity. So certainly this tissue culture is like one of the say boon for this agriculture field. So in coming days, the agriculture will mostly depend on the agriculture biotechnology and tissue culture. Second is like you, if those who are aware may be knowing, like cannabis in US is being legalized, US and Canada. Now they are getting a very big challenge for the cannabis strain, which can give them a proper yield of the THC and CBD. These are the alkaloids of the cannabis. And based on this yield, basically their business depends. So better yielding CBD and THC cannabis are grown using the tissue culture technology itself. So I'm consultant and an advisor to some companies to help them improve the strain of the cannabis. So like this, all because agriculture is crop based. So in crop, all biotechnology can be, plant tissue culture can be implemented and can give you better yield, disease free plants, and good return to the farmers and the business people. Yes, please.
Next question is, do you also provide a consultancy service?
Yeah. I'm consultant to 3-4 companies. We right from the designing the lab, procuring the instruments, and then training the manpower, preparing the project report and execution. So A to Z, I provide consultancy. I've established some laboratories and I'm still doing.
Okay. And for how long you have been into this field?
25 years.
Okay. Okay. Okay, Dr. Ajay, I don't see any more questions from our participants. So now we have come to end of question round. On behalf of agricultureinformation.com, we like to thank you for a detailed presentation and also showing the video of the lab. And we also like to thank all the participants for joining this meeting. The meeting will now be closed. And for your information, I've shared your contact number and email address with our participants. Just in case if they have any doubts, they can directly reach out to you.
Fine. Thank you so much.
Okay. Yeah. Most welcome. You're most welcome. Thank you.